Hello, today I want to demonstrate DNA replication. Hopefully you've read through the textbook chapter, you've watched those previous videos, so this demonstration makes a little bit more sense. You've got some familiarity with the concepts. So in order to draw and show DNA replication, the first thing that I'm going to do is I want to draw my old or original parent or template strand of DNA. Now I apologize that we've got essentially 4 different terms for what's the same thing. I really like the term old or original strand of DNA because when we do DNA replication, the whole point is to copy the DNA molecule to make, for one DNA molecule to make 2 genetically identical DNA molecules. And so by using the term original DNA, at least in my mind, it helps remind me that all of the new DNA molecules are made off of that original piece of DNA. But alas, videos and textbooks all use different terminology. So this in black is my old, original, parent, template, DNA. Now what I want to label here on my original old parent template DNA is I want to indicate directionality of each DNA strand. So arbitrarily, I'm just going to assign this side as being my 5' end, and if I call this my 5' end, I follow my wiggles. Down here will be my 3' end. If this is 5 over here, my opposite piece of DNA goes in the opposite direction. So up here will be my 3' end, and I follow those wiggles. Down here will be my 5' end. Now the reason why I'm doing this is because the directionality of my original DNA molecule is going to determine which way my new pieces of DNA are going to be made. It's going to determine the direction of synthesis for my new strand of DNA. Now there are 3 main steps to DNA replication. Helicase is going to unwind this original piece of DNA. DNA polymerase is going to build the new strand, and ligase is going to come in and fuse together those sugars and phosphates on our new strand of DNA. So here I'm going to draw helicase. And helicase is an enzyme, it's a protein, and the way we often draw proteins is we draw them as a cartoony blob. So here we have our helicase. And our helicase enzyme is going to be unwinding our old or original DNA in this direction. Where helicase is doing this unwinding is also known of as our replication fork. And the reason why we call this a replication fork is because we have this kind of tined look to the structure as helicase is unwinding in this direction. The next enzyme that's going to come in and do some work will be DNA polymerase. So I'm going to draw DNA polymerase as this kind of red blob. DNA polymerase is going to be building our new strand of DNA. It is going to be using complementary base pairing in order to build and make that new strand of DNA. So every time it reads an adenine on our original piece of DNA, it's going to put a thymine in our new strand of DNA. Every time it reads guanine on the old piece of DNA, it puts cytosine. Every time it reads cytosine on the old DNA, it puts guanine. And every time it reads thymine on the new piece of DNA, it will put adenine. Once DNA polymerase has built and made this new strand of DNA, our next enzyme that will come in will be ligase. Again, I'm drawing it like a cartoony bubble. I'm only drawing it on the new strand because as the new strand is being built, we've got those nucleotide monomers, the sugars and phosphates bonding together. we want to make sure every single sugar and phosphate is bonded tightly together. It is that sugar-phosphate backbone, drawn as a single line here, that gives this entire DNA molecule stability. Now, we don't need to go back and do anything to our old strand of DNA. Those sugars and phosphates have been bonded together for a long time. We know that this is stable, we know that this is fine, but ligase will come along and make sure that this strand of DNA is completely fused. All of those sugars and phosphates are tightly bonded together. A couple other terms that we need to label here for DNA replication. Where DNA polymerase is doing all of the work, this is going to be our origin of replication. And the final thing we need to label is our replication bubble. So this is half of a replication bubble. We have to imagine that this connects and continues on down below the dry erase board. We would have a second helicase, DNA polymerase, and ligase choo-choo train going in the opposite direction. So here we have half of a replication bubble. So when we look at DNA replication, helicase unwinds, DNA polymerase is building. DNA polymerase is building this new strand of DNA from the 5' to the 3' direction. It's using complementary base pairing. Where DNA polymerase is working is our origin of replication. Once everything is done, once DNA polymerase is done building that new strand, ligase will come along and fuse together the sugars and phosphates. And once both strands of our DNA molecule have been completely copied, that's when telomerase will come in and add those little shoelace caps, those little telomeres to the end that will help keep our DNA molecule from unwinding and help determine the lifespan of this DNA molecule and potentially even trigger when our cells should die. If you have any questions, email me.